When I heard the news this morning, I was stunned just because I wanted to know how T cells work. Ended up helping people with cancer. I'm a basic scientist, and the fact that I got it for physiology and medicine is so gratifying to me. I think my prize will impact MD Anderson by really helping work there. It's making a big difference. The teamwork there and the spirit that people have and the desire to help cancer patients is impressive. I've been a cancer patient myself. It's really special. As far as I know, first Nobel Prize has ever been given for cancer therapy, and I hope that it will create optimism. There is hope, and we're still working and trying to make it better. There are already many types of cancer that respond, but some that don't. And that's the big challenge that we face now, is how do we get at those?
To Jim. Yeah. Jim has been at MD Anderson now since 2012. He really came back to MD Anderson. He's from Texas. He grew up in Texas. He grew up in Alice, Texas. He used to tell me that as a kid, he would come from Alice to Houston in the summers to cool off. So we're really happy to have him at MD Anderson, where he is continuing to do cutting-edge research and applying now his findings to many other kinds of cancers, understanding immune resistance and how to improve these therapies yet further. Today is such a momentous occasion for Jim and his family. Pam Sharma, who also herself is an outstanding clinical investigator, immunotherapist, and son Robert Allison, who's here. So congratulations to the whole family. On behalf of the faculty and staff, our president, Peter Pisters, from MD Anderson, we congratulate you on this much-deserved honor. We're very proud, but also very happy for all of the patients that you've helped, all of the patients that you've helped now, and the patients in the future that will further be helped by your tremendous research. So again, congratulations, Dr. Allison.
Thanks, Patrick, for those kind words. I want to start off by saying I'm still in sort of a state of shock, and this is all sinking in. So I'll try to make some sense while I'm talking here. But it's really wonderful this morning to wake up, and my son called me at 5:30 and was the first to let me know that I'd won this prize, and I want to thank him for that, and Pam Sharma, my partner in life and in science. We work together now trying to improve immunotherapies. Also want to thank a long line of students and postdoctoral fellows and colleagues at MD Anderson and at UC Berkeley and at Memorial Sloan Kettering over the years that have helped bring this work to where it is, and also the patients who, especially the early trials, who put themselves at some risk when taking a drug which could potentially unleash the immune system to eliminate cancer, but also possibly to do harm. And luckily those effects are not all that serious, or at least can be managed. And so it's really turned out to help quite a number of patients. And I guess the reason I'm really thrilled about this is I'm a basic scientist. I did not get into these studies to try to cure cancer. I got into them because I wanted to know how T cells worked. And once we showed that this molecule CTLA-4 was the first cell-intrinsic negative regulators of immune responses, it immediately occurred to me and some of the people in my lab that maybe we can use this to unleash the immune system to attack cancer cells. And so I'm lucky enough, as a basic scientist, to see my work actually end up 20 years later, actually really helping patients. And I think that I want to say this modestly, but I think that this award, I was told by the Nobel Committee when I was called this morning that this was the first prize they'd ever given for cancer therapy. And so they've given some for cancer, the causing cancer before, but this is the first time for cancer therapy. And I'd like to just give a shout out to all the patients out there who are suffering from cancer, to let them know that, you know, we are making progress now. We can get durable responses in a fraction of patients with many, many different kinds of cancer. And of course, we still have a lot of work to do. There's some kinds of cancer. Well, first of all, it's a fraction of patients who respond. In melanoma, it's about 60%. We need to get that up as close to 100% as we can. With other cancers, it's somewhat lower. We need to get all of those, the fraction of patients that respond, and also bring these therapies to some kinds of cancer that have not yet responded, including glioblastoma and pancreatic cancer, the other things that you hear about in the news all the time. And we're trying to do that at MD Anderson in the immunotherapy platform there that Pam Shorman and I run together, where we're trying to understand mechanisms of what goes on exactly in patients, both when it works and when it fails, and understand the reasons for both, so that we can do rational combinations in the future to bring the benefit to more patients. So we've still got a lot of hard work to do, but the optimism comes from the fact I think we know the basic rules now. We've just got to work hard and learn the details and develop more personalized treatments for the majority of patients. And like I said, the good news is I think there's optimism to think that we can do that, but it's going to take a while. So I guess I'll stop there and take any questions that you might have. Who's okay? Right.
I wonder if you could just describe your mindset at the time, and what was it that got you up every morning and going into the lab when it was just purely for understanding some basic things?
Well, the dirty little secret, I think, of most scientists is that what really motivates us at the end of the day mostly is the desire to know something that nobody else knows, and hopefully something that's important, to be the first people to really figure out something complex. I mean, ever since I was a kid, I like to solve problems, and I think that's what keeps scientists going, is, you set yourself towards a task. I mean, when I started working on T cells, nobody knew how they worked at all. Didn't know the structure of the molecule that recognized the things that they would attack, didn't know the molecule, didn't realize that actually that there were additional molecules that were required to both, accelerator pedal and brakes, and then other modulators down the road. So it was really that kept me going most of the time, because science is a long and frustrating road to get in. There's no instant gratification of it. It's just the whole nature of science is trying to prove your hypotheses wrong. The better you do that, the better you're doing. And so you've got to be, comfortable with a lot of failures to get there. But I've had, a lot of cancer in my family, and my mother passed away from lymphoma when I was 10 years old, and she'd had radiation therapy, and one of her brothers died of lung cancer and had chemotherapy, and I saw the ravages of those kinds of treatment. And as I got into immunology, I always had the hope, and read a lot of papers, particularly from Lloyd Old, who I was lucky enough to have as a mentor, who in the early 60s began working on using the immune system in animal models. So it was always in the back of my mind. It wasn't the reason I was doing the work, but whenever I could, I would take a rest, from thinking about the problem and look at our data and say, what does this tell me that I could use to treat cancer? And that was one of those moments when we figured out that CTLA-4 was the brakes on the immune system. I said, well, let's just disable the brakes and see if that will allow the immune system to attack cancer. And it did. And so we did the animal experiments, and several years later did the early clinical trials. And we have patients now, actually one patient from the phase 1 trial in 2001. They got a single dose of N-acetyl-A4. A patient with metastatic melanoma is still around almost 19 years later. So these are very durable responses, and again, they're in a fraction of patients, but that's what we got to work on now. We know we can do it. We just got to learn how to do it better. Rather long-winded answer. Sorry about that.
Any other questions?
Yes, I'm with Agence France Press AFP. What was your reaction when you got the phone call? Tell us where you were.
Well, I was in the hotel that the Cancer Research Institute, which I've been associated with for many years, was having a major meeting up in Times Square with it's an international meeting with people from immunological societies all over the world, and well, Europe and the United States anyway. But so I was at that meeting. Pam Sharma won an award there, gave a talk yesterday, and so I was in the room, and my son called at 5:30, and then pretty soon I started getting calls from a lot of other people, and then a bunch of people came to our room, to the door, and beating on the door at 6 in the morning with champagne, and so we had a little party early this morning. It was really great with my friends, all of whom work in this field in one way or another, at the meeting. So it was tremendous.
Well, what was your emotional reaction to winning the Nobel Prize? I mean, when
Yeah.
did you speak to the committee? Did they call you?
For some reason they had trouble and didn't reach me until after I knew about it from my son and my friends. But they finally got me, and we talked about it. And that was when the committee member, said we've never given one for cancer therapy before. I guess it still, as I said, it still hasn't completely dawned on me, except that I'm a basic scientist, and to have my work really impact people is just one of the best things I can think about. I think it's everybody's dream, and I've been lucky enough to, do work that is benefiting people now, and I'm still trying to, figure that out. It's quite a shock to learn this at 5:30 in the morning. So, but it's as I said, I think it's every scientist's dream to have, first of all, do fundamental work that's important, but then to have it translate into helping people is just all that much more better. And have it recognized by the Nobel Committee is, I think, going to bring attention to the issues, the importance of basic science, and that there is hope for cancer patients.
Hi, okay. My name is Miho. I'm from Japanese broadcaster called Nippon TV. Congratulations on receiving this amazing award. And I just wanted to ask you actually about Professor Honjo, who also received that award today. Could you tell us, if you've met him, if you worked with him?
Yes, I know Tasuku Honjo very well. We've worked together. Well, not really worked together, but known each other and discussed science, and not on this, but on other topics since the early 80s, actually. And so he made this discovery of this molecule PD-1, which was the second checkpoint discovered. And then about the time that, we had a problem understanding why acetyl-A4 only worked in about 20% or so of patients. And so there are a number of possible explanations for that, but of course the most interesting one was that it's not the only checkpoint, that there's another one, and Honjo, his work brought PD-1 to the fore, and now we know those two together are extremely powerful. acetyl-A4 has a response rate in melanoma by itself of about 22%, but if you add PD-1 blockade to it, it goes up to about 60%. And so it just helped, show that this approach was good, and to broaden the impact and broaden the possibilities for helping patients.
Could you actually, sorry, one more question. So could you give us any comments or message to him? If you can?
Sorry, the last
Could
one?
you give us any message or comment to Professor Honjo?
Yeah, just give him my best and say I look forward to seeing him in Stockholm in December. We shared the Tang Prize several years ago, so we go back a little bit on this too.
Yes. Malcolm Ritter from the Associated Press. Just to go back to your story of how you heard, it's not clear to me. You said there was your phone rang at 5:30, but you didn't answer it, or there were other calls. I'm a little
No,
confused.
no. The phone rang at 5:30, and it was my son calling me,
Oh,
and spoke just for a few seconds because there was a call from Sweden, and I thought, oh well, because he'd already told me that that I'd won, but it was a reporter from Sweden, so.
oh.
And I talked with him briefly, and then my friends all showed up, and we started celebrating. And it was a while later. I still don't understand why they, the reporter had my number, but the committee didn't. But anyway, they finally found it and called me. I don't know what time it was, 7 or something like that.
So you heard the news from your son.
Yeah.
Okay. Thank you very much.
Kieran Weiser, Fuji TV. You seem like a very modest person and not really wanting to take a lot of own personal merit in this award or own personal boasting. As a scientist, how does it feel to be able to share this award with a fellow scientist, let alone a friend like Dr. Honjo, who you respect very much?
It feels very nice. As I said, we did it, the CTLA-4 work. It was kind of just, I don't want to say one-off. That's not quite what I mean, but it was a singular thing, and one of the worries was that it was going to be stuck at that 20-something percent, and I'd known Hanjo from the early days when we were both trying to figure out the T-cell antigen receptor structure, which we had done, and we considered working together to clone the genes for a while, but that's when I first met him. But he's done a lot of work on antibodies, a lot of areas of immunology he's worked on, and made many really important contributions to fundamental immunology, particularly in B cells, but more recently in T cells as well. So it was great to have him come along and identify another one of these molecules, as I said, that can broaden the applicability of the general theme of checkpoint blockade to cancer therapy.
One more question, if that's okay. What do you think makes you and Dr. Honjo such great friends as both colleagues, but also away from the workplace?
Away from, sorry?
Things that are unrelated to work? Do you have any special
Oh,
personal
yeah.
memories?
Yeah, a little bit. I mean, not all that much because he's in Japan and I'm here. But we've met particularly at scientific meetings. We've never golfed together. He plays golf. I don't play golf, so so that's never happened. But we've had a cordial relationship for, like I said, 30 years or so.
Sure. Thank you.
Do we have any more questions in the room? Otherwise, we'll take some from the phone. Oh, yeah. Yes.
Oh, yeah, Bob Langworth with Bloomberg News. Congratulations. I just wanted to ask, why do you think if scientists have been working on immunotherapy, particularly the vaccine area, for many, many years before your work, why were companies and cancer doctors not focused on this checkpoint blockade area until you did your work? Just kind of explain your kind of perspective and why that was less researched.
Well, I think the idea of using the immune system to treat cancer goes way, way back. The problem was it started being applied before we really knew how it worked. And so, for example, with cancer vaccines, I think that looking back on it in light of what we discovered with CTLA-4 is that, after the first vaccination, since the CTLA-4 is turned on by the antigen receptor stimulation, you vaccinate, you're really increasing the signal through CTLA-4. So every time you do it, you're turning the immune system off. You're beginning to, dampen the signals down, and people just didn't realize that. And I think that another thing is we just don't know how to we still don't know the proper way to vaccinate, to get therapeutic responses. Obviously, we could do a great job against microbes, but in terms of really instantly mobilizing the immune system to go after something like cancer, we still have a lot to learn there. But basically, I think it was a lot of the early immunotherapies were they just started with insufficient knowledge to know the details of the circuitry that we were working with.
One last question here from in the back. Just in the simplest way you could explain it in the shortest time period, what for average people, what does your and your colleagues' discovery mean for the future of people who may be suffering from cancer?
I think that what we've learned, again, to put it very simple, is that there are built into the immune system these inhibitory circuits that stop the immune system at a certain point so that it doesn't hurt normal tissues. And that also keeps it from attacking tumor cells as effectively as the T cells might. And so we found a way to temporarily suspend the brakes and let them take off. So after many years of resistance, I think the world, the cancer field has begun to accept immunotherapy now as the fourth pillar, along with radiation, surgery, and chemotherapy, of cancer therapy. What we have to look forward to is that, unlike the other three, immunotherapy can be used in combination with the other three. And I think that what we're looking forward to is combinations in the future, not just of multiple checkpoints, but of checkpoints with radiation, checkpoints with chemotherapy, checkpoints with genetically targeted small molecule drugs. To really, I think that immunotherapy is going to be part. It's not going to replace all those others, but it's going to be part of the therapy that essentially all cancer patients are going to be receiving in 5 years or so. And they're going to be curative. They're going to be curative in a lot of patients.
Move to instructions for the folks on the telephone.
At this time, if you would like to ask a question, please press the star and 1 on your touch-tone telephone. You may withdraw your question at any time by pressing the pound key. Once again, to ask a question, please press star and 1 on your touch-tone telephone. And we will pause for a moment to allow questions to queue.
Have some water.
We do appreciate your patience, and we will take our first question momentarily. And we will go ahead and take our first question from Ivan Perone with AFP. Your line is now open.
Dr. Allison, can you talk about the other pillars precisely that you mentioned? How important do these conventional therapies remain? And what should patients think and tell the doctors about? Should all the funding go to these areas, to immunotherapy as opposed to all the therapies?
Yeah, well, first, as I said, I think that there is still a place for the for the more conventional therapies. You know, there are some cancers that we don't have, you know, so far immunotherapy hasn't worked in. As I mentioned, glioblastoma and pancreatic cancer, those are big challenges. But the whole process of getting the immune system going after cancer starts with some tumor cell death. And that alerts anyway, without making it complicated, that causes inflammation. That starts a whole cascade of immunological events. And so if you can kill cancers with radiation, or you can kill cancer cells with chemo, what we know now is that the traditional way of giving high doses of chemo or high doses of radiation and trying to kill that last cancer cell, every last cancer cell, is no longer the goal and should not be the goal of those other therapies. You can moderate their use and just try to kill enough kill enough tumor cells to initiate the immune cascade, and then come in to finish the tumors off by activating, sustaining T cells. And so, you know, but I don't think all the funding by any means ought to go into that. I mean, we know that with the small molecules, for example, those drugs have been developed with in vitro assays and then in xenografts, tested in xenografts of human tumors growing in mice that don't have immune system. So in most cases, we have no idea whatsoever what those drugs do to the immune system. But we need to know that. if we're going to begin to combine them with immune therapies. The same for chemotherapy. We need to know, many chemotherapies can probably work. We need to know their effects on the immune system, though, and if they are immunosuppressive, to come up with staging, dose scheduling that allow them to be used together. But I would say that what we need to do is to continue to fund basic research, because that's where these ideas came from, not from trying to kill cancer, but from trying to understand how the immune system works. And if everything's translational and targeted on a given area, pretty soon there's going to be nothing to translate because there won't be any new ideas. So the big jumps come from the basic sciences, and I think we got to keep that in mind always. We can't just say everybody's got to be trying to cure a disease. It does not work that way.
For James, I apologize. I just arrived. You probably already said what I'm going to ask you, if you don't mind. My name is Fabio from Brazilian Global TV. Just would like to ask you a few words about how you're feeling by getting this prize, and also what kind of change did your findings contribute to the cancer treatment and perspective to the patients all around the world?
Yeah, well, I guess as far as I'm feeling, I'm still kind of in a state of shock, actually, from getting up this morning and learning this. But the thing that gratifies me is I do consider myself, well, anyway, I used to be, maybe not anymore, but a basic scientist who was just trying to learn about the immune system and happened to come across something that we could exploit due to the help of a lot of really, really bright students and fellows and everything. I think what this means to cancer patients is, if I dare use the word cure, which I know is a very dangerous thing to do, but we have patients now that are 18 years out since they get one round of therapy and they're 18 years out, 14 years out, whatever. And I think those patients can be considered cured. And I think that that's the hope that these kind of therapies work, because the immune system, after all, differs from conventional therapies in several ways. One is that once you've got T cells, you've got them for the rest of your life, and so they don't go away. If you give a chemotherapy drug, it's gone in hours. And so you really have to keep giving it until you kill every last tumor cell. You don't have to do that with the immune system. The cells will be around and can keep coming after the tumor and changing as the tumor changes. To recognize new antigens on the tumors. The problem with cancer is the tumors are so heterogeneous because of the genomic instability that they're very hard to treat. But that makes them, just easy targets for the immune system, because the immune system loves heterogeneity, and the more antigens, the better. The more mutations, the better. And I think that, again, what this offers, we've still got a long way to go. We're curing patients now. The idea is to really learn in detail how it works and bring these benefits to more patients. And it's a challenging thing, but what we know now that we didn't know 10 years ago is that it can be done, at least in some patients. So we've got to grasp on it, and we just need to, keep working.
Do you already know how many patients already benefited from this therapy?
Not really, but I think hundreds of thousands have been treated, so it's tens of thousands, I'm sure. I don't know. I've been trying to ask Bristol-Myers Squibb, who, helped us get anti-CTLA-4 on the market, and nobody seems to know, but it's a big number. 4 years ago, for example, there were 5,000 patients at least who they had 10 years follow-up on, so the real number is somewhere way above 5,000, just with ipilimumab. And then with PD-1 and the new combinations and stuff, it used to be that this therapy was given to late-stage patients who had no other options. Now it's the first choice for melanoma, for example. A patient starts on immunotherapy. Rather than chemo or even the genomically targeted drugs. The only patients that get those are the patients who fail immunotherapy now, and I think that's the way many, many cancers are headed.
Okay, we have one more question from the phone.
So we will take our next question from Tina Safe with Science News. Your line is now open.
Hi, Hi, I was wondering if you can tell us a little bit more about why certain cancers seem to respond to these checkpoint inhibitors better than others do?
Yeah, there are several reasons. One is there seems to be an association with the total mutational load, the mutational burden. Those that have a lot of mutations, those we know now with the immune system, it's sort of the T cells in the case of cancer are preoccupied with targeting proteins that are made by the mutations that occur during the cancer process itself, not just the drivers, as they're called, that cancer biologists have been looking for, but has the potential to recognize all the passengers as well. So melanoma, for example, can have 3,000 or so mutations per cell. So that's 3,000 potential targets to see. But you get down, it ranges actually, it's a range from a few hundred to thousands. But if you get down to something like prostate cancer, there may be 30 expressed mutations. Now that doesn't mean that absolutely is unresponsive. We recently found in a study that we can see T cells that recognize mutations out of even a small number of mutations. But that's part of it. I mean, the more mutations, clearly, the more likely you are to get one or more that the immune system can recognize well. Another one is that a lot of tumors are what we call cold, or they're immunological deserts. There's no T cells in them, and so until you get the T cells in, you can't really do anything. We know now also, though, that CTLA-4 will drive T cells in tumors, but then it also induces expression of more checkpoint inhibitors, such as the ligand for PD-1. And so then you have to start thinking about giving combinations to deal with it. There's some new ones coming along. So, and then also in some tumors, it looks like they have active methods of suppressing, multiple methods of suppressing the immune system and protecting them.
Ladies and gentlemen, I wish you all good morning and very welcome to Nobel Forum for the announcement of this year's Nobel Prize in Physiology or Medicine. So I will first read the announcement in Swedish, followed by English, and we will then present some background to the prize and then open up for questions, of course. Nobelförsamlingen vid Karolinska institutet har idag beslutat att Nobelpriset i fysiologi eller medicin år 2018 skall delas lika mellan James P. Allison och Tasuku Honjo för deras upptäckt av cancerbehandling genom hämning av immunförsvarets bromsmekanismer. The Nobel Assembly at Karolinska Institutet has today decided to award the 2018 Nobel Prize in Physiology or Medicine jointly to James P. Allison and Tasuku Honjo for their discovery of cancer therapy by inhibition of negative immune regulation. Here we see the two new laureates. James P. Allison was born in Alice in Texas in the United States. He performed his prize-winning studies at University of California, Berkeley, and at Memorial Sloan Kettering Cancer Center in New York. He's now active at the University of Texas MD Anderson Cancer Center in Houston. Tasuku Honjo was born in Kyoto, in Japan. He performed his prize-winning studies at Kyoto University, where he's still active. I will now leave the word to Professor Claes Scherre, member of the Nobel Committee, who will describe this year's discovery. So please, Claes.
Ladies and gentlemen, let us start by a summarizing metaphor. It is all about interference with the brakes of our immune system as a means to unleash this defense and harness it to develop treatment of cancer. Cancer is a common term for many different diseases caused by uncontrolled proliferation and migration of cells. Eventually, cancer cells can spread to distant organs, so-called metastases, usually leading to death. Cancer represents a major medical challenge in the global perspective. There are 3 pillars in the treatment of cancer: remove it by surgery, eliminate it by radiation, or attack it with drugs that harm the cancer cells. Treatment has improved considerably during the last 50 years. More than 2 out of 3 are cured today. But note that these are average figures which conceal major differences between cancers in different organs, between early and advanced disease, and also between different countries. For many cancers, very few patients can be cured. Every year, many millions of people die from cancer. So novel types of treatment are desperately needed. There has been a huge investment in basic research with the aim to acquire knowledge as a platform for new therapies. One area was of particular interest: can immunity be harnessed to attack cancer? So let us look at our immune system. It protects us from infections. It comprises a great diversity of cells and molecules with different functions, and they can be found all over the body. A fundamental property of the system is its ability to discriminate between self and non-self, so that viruses and bacteria can be attacked. And indeed, innovative basic research in tumor immunology during the last 50 years have revealed a number of ways whereby cancer cells can also be recognized as non-self. This led to cure of cancer in many mice and also to successful treatment of some patients. However, it was impossible to develop efficient, generalizable therapies for clinical practice. Clearly, an important piece of the puzzle was missing, and this is what our laureates this year have solved. Now, let us take a closer look at the T cell, with the T appearing here, and we focus on it. In the 80s, scientists had discovered T cell receptors that bind to structures recognized as non-self. You see the structure as a dot in purple here. These structures can be picked up from cancer cells and be presented by so-called antigen-presenting cells. T cells that recognize something foreign with the receptor, as here, they get a signal to act, but this is not sufficient to get them going. Additional proteins on the surface of the T cells must also engage in the decision, acting as accelerator or gas pedal. They were called co-receptors, and the research field around them was termed co-stimulation. Many researchers contributed and observed that even more molecules were involved, but with quite the opposite function. They instead worked like brakes. And here they are in yellow, or at least one of them in yellow. And this one is called CTLA4, a complicated name, but please remember it. It will be very important. So when you pull the brake, it tells the T cell to hold back, stop. Allison was one of the researchers who observed that this molecule works like a brake. You can see that it interferes with the accelerator on the T cell. Now, some research teams started to explore this to treat autoimmunity, but Allison, he had an entirely different idea. He had developed an antibody to CTLA4, shown in green here. And if you block or release the brake with the green antibody, Allison reasoned, the consequence should be that the T cells are unleashed and become more active, and they should now hopefully attack the cancer cells. This was the hypothesis, and the key experiment was performed in Allison's laboratory at University of California, Berkeley, over the Christmas in 1994. And the results were spectacular. The mice who had been treated with the antibody, the yellow curve, were cured from cancer, while the control mice developed large tumors. This seminal study represents the birth of a new concept for immunotherapy, today often referred to as immune checkpoint inhibition. The brake acts in the checkpoint. Allison was now eager to apply it for benefit of patients. At first, there was only limited interest, but eventually a small biotech company took on the challenge to produce the human antibody, the first step on a long journey. A few years later, the first clinical trials were started, and one comprised 14 patients suffering from metastatic melanoma, a pigmented cancer of the skin. And this picture shows how a metastatic melanoma can present on the patient. Now, he started to treat the patients, and some of them showed striking responses. The tumors shrunk, they could even disappear. Other patients showed a more modest or no response at all. And severe side effects were noted in some patients, but the results were positive enough. And this started a long series of larger trials conducted by clinical colleagues. To cut a long story short, in 2011, the treatment was approved for inoperable metastatic melanoma. Allison had now established a novel concept for cancer therapy, but several questions remained. Could checkpoint therapy be developed to cure more patients, and could it be generalized to other types of cancer? Studies that would lead in this direction were already ongoing in a laboratory across the Pacific in Japan. Actually, they had been started several years before Allison's key discovery. So we cross the Pacific, we go to Japan, and we go back in time to the point where Allison had started. And you will remember and recognize the T-cell receptor and the accelerator. Enter our second laureate, Tasuku Honyo, who in 1992 discovered a new protein on T cells, which he called PD-1. And PD-1 is shown on yellow here on the T cells. He did not understand what the molecule was doing, but together with his colleagues at Kyoto University, he launched an ambitious, curiosity-driven research program to understand its function. Several years of research later, he could reveal a chain of molecular interactions, leading him to conclude that PD-1 also acted as a brake, just as CTLA-4, but in a different manner. You step on the brake, and the T cells holds back. In addition, Honyo had identified the ligand, the molecular partner of PD-1, and he found that it was expressed on antigen-presenting cells, but also on cancer cells. And now it became immediately evident to test whether blockade of PD-1 could also be used to unleash T-cell responses against cancer. So what happens when you release the brake like this? Honyo's and other groups immediately started to gather evidence for this. And here we are looking at one of the studies of melanoma, the skin cancer, and in this case it is spreading to the liver in mice. The control mice on your left developed multiple metastases in their livers, while several of the mice who had been treated with the antibody to your right had almost no metastasis at all. In this study, Honyo correctly predicted that anti-PD-1 treatment might complement the anti-CTLA-4 treatment, since the two worked via different mechanisms. He also predicted that PD-1 might give less side effects. Honyo now also turned to the pharmaceutical industry. An intensive program started, conducted by other clinical scientists, mainly in the US. Again, similar or even more dramatic responses were noted, not only in melanoma patients. And FDA approved the anti-PD-1 treatment for melanoma in 2014 and for lung cancer as well as kidney cancer in 2015. So now we have two treatments available for patients based on immune checkpoint therapy, and today we have even more. And it's time, I think, to look at some results in patients. These CT scans are from a patient treated with anti-PD-1, but they illustrate processes seen also with anti-CTLA-4 treatment. You can see the tumor marked by the red arrow in the lung. Note how it has shrunk dramatically to your right, and it's on its way to disappear at 4 months after initiation of the treatment. But please note also what happened shortly after the treatment had started, at 2 months in the middle. The tumor appears to be significantly larger than it was from the beginning, but this is not real growth of the cancer. It is due to swelling caused by the inflammatory response, with many immune cells entering the tumor. And this is called pseudoprogression and is observed quite frequently, and it caused confusion initially, since other types of cancer treatment usually results in an immediate response and shrinkage of the tumor. Let us finally look at an example of compiled results from many patients. These curves represent survival of treated patients with a certain type of melanoma. And you should note that just a decade ago, most patients with advanced disease like this would have died already after 2 years. Previous standard therapies resulted in long-term survival for few, 10% or less, and it would have looked like this. The curve compares 3 groups of patients treated with checkpoint inhibition. The release of one brake, CTLA-4 in green, release of the other brake, PD-1 in blue, and release of both in yellow. And we can appreciate that both treatments are working, anti-PD-1 in a higher proportion of patients than anti-CTLA-4, but please note the combination, where more than 60% of the patients are still alive after 3 years. This is a spectacular advance in the treatment of a solid metastatic tumor, and we can expect even better results in the future when these different checkpoint inhibitors will be combined with conventional treatments. So, in summary, Allison's and Honjo's discoveries have added a new pillar in cancer therapy. It represents a completely new principle because, unlike the previous strategies, it is not based on targeting the cancer cells, but rather the brakes, the checkpoints of the host immune system. The seminal discoveries by the 2 laureates constitute a paradigmatic shift and a landmark in the fight against cancer.
Thank you very much. So if we could have the lights a little brighter here. So I would like to open up for questions. I would like to mention that to our help here we have some distinguished experts, all from the Nobel Assembly and the Nobel Committee. Dr. Jonas Berg, who is an expert in oncology, Ted Smith from the Nobel Committee, Dr. Anna Wedell, the chair of the Nobel Committee, and of course Klaus. So please, if there are any questions, here we have one.
Hello, my name is Fred from China's National Xinhua News Agency. I've got a question for the experts. So this year's Medicine Awards is given to the new cancer treatment. So what are the significance of this award? Are we saying that the cancer treatment has been a huge challenge nowadays for the human being? What are the significance for this award this year?
Yep. So this type of cancer treatment is very interesting because it works in a completely new fashion. All previous cancer treatments were essentially directed against the tumor cell, but this particular treatment is directed against the immune system. And one interesting aspect is that it works on many different types of tumors, not only on one particular tumor, but on many different types.
Here we have one question. Can you please wait for the microphone? Okay, we'll start with one up here and then your. Okay. Please go ahead.
Okay, I have a question about James Allison anti-CAR-T4 therapy. I know that another excellent scientist from Yale University, Lipin Chen, he's also a highly eligible candidate for this. Just would like to know the reason that he's not being selected. Thank you.
Claes, do you want to respond to that?
The question was about another scientist who is not given this year's award. Now, let me first say that the Nobel Committee divides the world, or the people of the world, in two groups. The ones that have received the Nobel Prize in Medicine and the ones who have not yet received it. And we love to comment on the ones who have received it, but prefer to refrain from comments on the others. Having said that, I would like to add that, as I tried to imply in this presentation, many scientists contributed to this whole field. And if you want to read about the contributions of different scientists, many of them very, very smart and clever, who contributed a lot, we refer you to the advanced information that is, I think, is now out on the Nobel web, where you can read about all the contributions of many other scientists, which were also important, but these two stand out.
Okay. Here's one question.
Yeah. Thank you. I'm Shifei Axel Song from Green Post and China Radio. I'd just like to ask you that when we talk about this immune checkpoint therapy, does it mean that it is a method that narrow down the point to check for the cancer treatment, or how better it is than the previous ones? Thank you.
Could I ask Jonas Berg to answer that question?
The effects of these checkpoint inhibitors are quite remarkable because they really define a new concept where you release the brakes. Previous types of immune therapy, they were really trying to make use of the immune system by stimulating it and enhancing its capacity. But here, in that concept, you didn't release the brakes, and for that reason, those therapies were not as successful as these therapies. And the fundamental thing also is that these therapies, as we are discussing today, they are apparently and very nicely working in previously quite therapy-resistant tumors, as mentioned here, malignant melanoma, different types of lung cancer, to some extent renal cancer, to give some examples, while quote-unquote simpler cancer, which have been sensitive to endocrine therapy, cytostatics, radiotherapy, etc., the effect by the conventional therapy has been quite good, but for the tumors I mentioned, that present armamentarium up until today have not been that fantastic.
Yeah, what about the side effects?
Could maybe, who wants to answer that? Maybe it's best that you go ahead with that, Jonas.
The side effects are there, it's correct, but if you compare with other approved drugs or therapies for cancer, there are by all means always side effects. And if I compare the armamentarium, the new drugs we are discussing, the checkpoint inhibitors, the side effects are for almost all patients manageable, although they are in a scale up to 5. They are not rare with grade 3 to 4 side effects, but mostly of them, they are reversible. And it's clearly so if you look at the benefit, namely the effect on the cancer, and then you balance that versus the risk, there is no question that there is a clearly positive benefit-risk balance for these, for the therapy of the cancers we have discussed here.
There's one up here.
This is from Nordic Chinese Times. I have a question for I would like to thank you for these two great scientists. But we have now run thousands of clinical trials with PD-1 and CTLA-4 and with combination therapy. So we even run out of patients. So what's the next future or rational design for this combination with this checkpoint therapy? Thank you.
Ask Anna to respond to that.
Yes, I think what's most promising now, we have now a completely new tool to add to the toolbox. So it has sort of revitalized the whole area. So to find the right combination for the right subgroups and even for the right patients and go into a more individualized therapy will be a very important process. There are very many studies, of course, ongoing doing that. So there will be more concepts, more checkpoint inhibitors most likely, but combining what we already have also with conventional therapy, there's a huge benefit to gain there, I'm sure.
Do you wanted to add something?
Yes, I can add that this is a very active field. So that paper last Friday, 2028, FDA approved yet another anti-PD-1 antibody, which is called Cemiplimab. I can also add when it comes to the side effects, the adverse effects, that there are differences. So the latest drugs directed against PD-1 and PD-L1, the ligand and PD-1, have fewer side effects than the first candidates that came up. Those directed against anti-CTLA-4.
The powerfulness by combined therapy, I can mention triple-negative breast cancer in a small randomized phase 2 study. If you just give chemotherapy in 20%, 1 in 5 patients, the tumor totally disappear in the areas which were there before therapy. If you do the combination of chemotherapy plus a checkpoint inhibitor, in 6 out of 10, 2 out of 3 patients in reality, the tumor disappeared. So it was a dramatic improvement from 20 to 60% in this very early study, demonstrating that combination for some patients could be quite useful using the conventional therapy strategies.
Mr. Pellman, David Keaton from the Associated Press. This is a question for you. We saw earlier a photo of you speaking on the phone to the winners. Could you tell us a little bit how they reacted when they heard the news that they were this year's winners?
Yeah, well, what you did see was speaking on the phone with Dr. Honjo. So I didn't get in touch yet with Dr. Allison. He might know it now, but I never got hold of him. Dr. Honjo sounded extremely pleased. I have a little bit difficult time to guess if he had some expectancy of this news, but he seemed rather surprised, and he expressed very clearly how satisfied he was, also of sharing the prize with Dr. Allison. Are there any other questions? So with that, I think then, if there is no one else who want to ask anything, that we can conclude this press conference. And there are some of you who asked for interviews, so we will try and satisfy all requests as quickly as we can. So thank you very much for coming. Thanks.
Tá longe, só quando a gente já tiver velho. Mas você vê, quando que ele começar a sair, né? É. Foi porque eu pensei, né? Mas quando que ele começar a Ah, os papers são mais ou menos recentes. Você tem lá 4.996.
Professor Claes Kärre, member of the Nobel Committee, we have just heard that this year you're awarding the discovery of checkpoint inhibitors. Could you please explain what this is?
Well, first of all, this is a treatment against cancer, and it's a new principle for treatment because unlike previous treatments where you target the cancer cell, this treatment targets the host's immune system, the patient's immune system, and it does so by releasing or blocking the brakes of the immune system, and when you release the brakes, things go faster and we have a more strong response against the patient's cancer cells.
At least one of the laureates' names, James Allison, he has been mentioned for quite a while. How come you are awarded the prize this year? Why now?
Time is ripe. The first approved drug based on this treatment came in 2011, and patients have been treated for several years now, and we can see the long-term outcome, and it's very convincing, and we consider it really a big step in the fight against cancer.
It was quite complicated, different cells and different receptors, but in order to describe this prize in an accessible way, what kind of metaphor would you use?
I would like to use the brake metaphor, the brake pedal that you have on a car, for example, where you can step on it and everything stops, and you can release it and you get things going. That's the best metaphor.
This is a new kind of cancer treatment. What cancer patients can be treated using this method? Are there any limits?
The most dramatic responses are seen with a particular type of skin cancer, malignant melanoma. And I'm sure most viewers know what this is. It's a pigmented cancer on the skin. Other cancers that respond, at least to PD-1 treatment, are lung cancer and kidney cancer and lymphoma, and many other cancers actually also. So it will be very exciting to see in the future exactly how broadly this can be applied.
Personally, what makes you enthusiastic by this prize?
I myself am an immunologist, and I have actually worked in tumor immunology for many, many years, and there has been a lot of interesting new discoveries in tumor immunology where we revealed basic mechanisms, but they were never sufficient to be able to cure cancer in patients. They were sufficient to cure cancer in mice, which is somewhat easier, and all the other previous attempts to use the immune system, such as vaccination or giving cells, they worked in some patients, but you could never generalize it to many patients, and this is what is different about this one, and it makes a big difference for certain patients. Some patients survive and are cured, and some patients get 4 years rather than 1 year, and that is quite important.
Now, if we turn to the laureates, James Allison and Tasuku Honju, who are they?
I have met them, but I don't know either of them very well. But Professor Allison is a basic immunologist. He's not trained in medicine, but in biology from the beginning. And he became interested already when he was a small boy, and his father gave him a chemistry kit. And he has had a splendid career in basic immunology. He made many important discoveries before this one, and he has been in many different centers in the United States. Berkeley was mentioned here, but he has also been at the Memorial Sloan Kettering Cancer Center in New York, and in MD Anderson in Texas, where he is now. I'm also told that he plays the harmonica in a blues band called the Checkmates. Professor Honjo is also a very distinguished basic immunologist. He's actually an MD, so he has medicine training, but he has also contributed very important discoveries in basic immunology before this one, and he was not in cancer immunology from the beginning, similar to Jim Allison, who also was not in cancer immunology. But these basic scientists made their discoveries and persistently followed them through and developed these fantastic treatments then. I'm not sure whether Professor Honjo plays any instrument, but I know that he plays golf and is very interested in art.
When they were doing this discovery, were they looking for this, or was it a surprise for them when they found it?
For Professor Allison, I think he was working with the basic regulation of the immune system, like many others, and he did not discover the molecule that he was working on, but he realized its potential. So he rapidly tested an idea that nobody else tested. I don't know how expected it was for him to get into cancer, but he did. Professor Honey, I'm sure, he did not think of cancer at all in the beginning. When he looked and found his molecules, he was doing completely different things and actually probably had ideas that it was doing something completely different. But as he went on and he gradually revealed how this molecule worked, he understood with inspiration from Allison how this one could add and even become even more efficient.
And finally, if you were to do an elevator pitch on this prize with the goal to simply convey the overall concept in an exciting way, what would you say then?
These scientists have discovered how you can block the brakes of the immune system so that it goes much faster and acts in a much more intensive way and thus can attack cancer cells, so we can cure cancer with it.
Excellent. Thank you very much.
Thank you.
We know now, based on studies of thousands of patients that have been treated, that about 1 in 4, 1 in 5 that get a single treatment will be alive 10 years later. I mean, they're basically effectively cured. I'm not an MD, I'm a scientist, so I have to work with the doctors to design what we're doing in the clinical trials. When you get it together and everybody's playing together, it's really special.